Common name, dragonflies and damselflies. Scientific name, Insecta, Odonata. Introduction. The order Odonata contains the dragonflies and damselflies and is one of the most popular insect groups. Odonats are popular with both the amateur and professional because they are large, colorful, easily observable and have exceptionally charismatic behaviors. In recent years, dragonflies in particular have been popular with birders as many dragonflies rival birds in wingspan, color, gregariousness, and predictability. As a result of their popularity with the public, they have become the focus of many conservation efforts in North America, Europe, and Asia. Odonata was until recently composed of three suborders: Anisoptera, commonly known as dragonflies, Zygoptera, commonly known as damselflies, and Anisozyoptera, as the name denotes, a morphological composite of the previous two suborders. However, the suborder Anisozyoptera has been abandoned, as current research shows that Anisozyoptera is not a natural group, and is paraphyletic. Wren 2003, Lohman 1996. Thus, the group has been combined with the suborder Anisoptera, which does form a natural group in a new suborder called Epiprocta, Bechley 1996. To facilitate the discussion of North American odonats, it is useful to use the names Zygoptera and Anisoptera when discussing differences between the damselflies and dragonflies, as there are no extant Anisozyoptera in North America. Dragonflies, Anisoptera, make up the more species of the two suborders and are much more easily observed than their dainty relatives, the damselflies. They have large eyes that take up nearly the entire head surface and when not contiguous are not as widely separated as within the damselflies, see the key below. They also have a very robust body structure to support a massive musculature that propels their large, broad wings. Dragonflies are unmatched as flyers and have a very agile, deliberate flight. Males are often territorial, defending oviposition sites from other males. Entomology and Nematology, FDAX, DPI, edis. Back to top. Figure 1. Dorsal view of an adult dragonfly from the family Libellulidae. Photograph by Seth Bybee, University of Florida. There are 6 families of dragonflies found in Florida, each of which is easily identifiable. These insects are commonly found around ponds and open bodies of fresh water and large blacktop parking lots. Perhaps parking lots resemble open bodies of water to odonats where they search for prey and mates. There are a number of species that disperse away from water for a time before returning with a mate to breed and deposit eggs. Damselflies, Zygoptera, comprise the more morphologically diverse suborder of odonats. In North America and particularly in Florida they have similar characteristics, which are eyes separated by more than the width of a single eye, abdomen much longer than wings, very slim body structure and a simple fluttering flight. Damselflies can often be quite spectacular in color, such as the ebony jewel-wing damselfly, Calopteryx maculata, found throughout the eastern United States and Florida wherever there are slow-flowing, shaded wooded streams. Figure 2. Dorsal view of an adult Calopteryx maculata, a damselfly. The species is in the family Calopterygidae. This image shows one of 2 general wing forms found within damselflies in Florida. Photograph by Seth Bybee, University of Florida. Figure 3. Dorsal view of an adult damselfly from the family Cynegrionidae. This image shows one of 2 general wing forms found within damselflies in Florida. Photograph by Seth Bybee, University of Florida. Away from the forest habitat one can find other families of damselflies. One of the most commonly encountered families is Cynegrionidae, which can be observed just about anywhere along the edges of water systems. A less common family of damselflies is Lestidae. This group is readily identifiable in the field by their behavior of perching with their wings held open, a characteristic more common to dragonflies than damselflies. Distribution. Odonats are found on every continent except for Antarctica. In particular, the majority of the families that comprise Anisoptera are broadly distributed throughout the world. Some species of dragonfly have vast distributions such as the blue-eyed darner, Rhinoaceana multicolor, which is found from coast to coast in North America, in addition to Central and South America. In contrast, many families of damselflies are narrowly restricted and several families have an extremely limited distribution. Florida is home to more than 150 species of odonats belonging to 3 families of damselflies, Calopterygidae, Cynegrionidae, and Lestidae, Daigle 1991, and 6 families of dragonflies, Eschnidae, Cordulagastridae, Corduliidae, Gomphidae, Libellulidae, and Petalouridae, Daigle 1992. They can be seen near any body of water or running stream, often sitting in sunspots breaking through the forest canopy along a river or pond, or patrolling the water's edge. Some species can be found in open fields far away from water while searching for prey or while migrating. Description. Eggs. Odonat eggs display a large array of shapes, from those that resemble tiny rice grains to minute mangoes. Odonats oviposit in 3 ways: endophytic within a plant, epiphytic, on the surface of the plant, and exophytic, on water or land, Corbett 1999. Generally, eggs oviposited endophytically are several times longer than wide while those laid epiphytically and exophytically are ellipsoidal to subspherical, Corbett 1999. Clutch sizes can be as large as 1,500, with some females depositing several thousand eggs in a lifetime, Corbett 1999. Eggs hatch 7 to 8 days after oviposition, but hatching can be postponed for up to 80 days, Miller 1992, and in one case 360 days, Sternberg 1990. Immature odonats, naiads. Immature odonats are sometimes referred to as larvae or nymphs, but here will be referred to as naiads because odonats are aquatic hemimetabolous, lacking a pupal stage, insects as immatures. Naiads live in most aquatic habitats. Some can even survive in salt water, Corbett 1999. All naiads are voracious predators feeding on everything from small invertebrates such as mosquito larvae to smaller vertebrates such as fish and frogs. Back to top. Back to top. Figure 4. Frontal view of a dragonfly naiad from the family Macromedae. This image shows the general form of dragonfly naiads. Photograph by Seth Bybee, University of Florida. Figure 5. Dorsal view of a damselfly naiad from the family Calopterigidae. This image shows the general form of damselfly naiads. Photograph by Seth Bybee, University of Florida. Naiads will molt 9 to 17 times before becoming adults, Corbett 1999. The number of generations per year depends on the species of odonate. Species at higher altitudes or in dry environments usually have one generation per year, while those in tropical habitats may undergo multiple generations per year depending on the availability of appropriate habitats. When naiads are ready for their final molt, they leave the water and crawl onto the bank or vegetation where they will molt into adults. Much like a caterpillar emerging from a chrysalis, they need to pump up their wings and allow their bodies to harden before they can be effective flyers. A newly emerged odonate is teneral, soft. A teneral dragonfly has glossy wings and the colors on the body are often pale. Several days after emerging and hardening completely, a dragonfly will have taken on the colors of an adult dragonfly. Adults. Identifying males and females is not difficult. Males will have what appears to be a pouch on the second and third abdominal segments that contains secondary genitalia. The actual male genitalia are found on the last abdominal segments along with a grasping structure used to hold the female while mating. The male produces sperm at the tip of the abdomen and transfers this sperm to the secondary genitalia where the female will have access to it. Females do not have secondary genitalia or grasping structures at the end of the abdomen but instead have a single genital opening and a small ovipositor at the end of the abdomen that will be used to oviposit her eggs. See above for types of oviposition. Normally, the male dragonfly is more colorful while the female will be a dull brown or gray. This is not true for all odonates. For example, both sexes of Calopteryx maculata are very similar in coloration with the exception of the female having a white pterostigma while the male does not. When odonates mate, they form what is called a mating wheel. The wheel is formed when the male grasps the female behind the head and the female raises the tip of her abdomen forward to come in contact with the secondary genitalia of the male. Odonates can often be seen flying in tandem in this fashion. Dragon colors. The nature and function of Odonata, dragonfly and damselfly coloration. Adult odonates, dragonflies and damselflies, exhibit a great diversity of colors which vary remarkably between species, between individuals within species, and throughout the individual's lifetime in some species. Here, we provide a summary of what is known about color recognition, and production of color including pigmentary absorption, structural reflectance, and fluorescence, in odonates. We also review the current understanding of the function of color in adult odonates, such as in signals during mate choice, in species recognition, and in predator avoidance, as well as in physiological adaptations to abiotic conditions. Finally, we provide some directions for future research. Eye and pterostigma color, coloration at different life stages, UV color, phylogenetic analysis of color evolution, color and hot climate patterns, and standardization of color recordings. Given how easily they can be marked and tracked, odonates are exemplary animals for field and laboratory research. Therefore, unraveling the physiology, evolution, and ecology of odonate color can provide significant advances, in general, to understand insect color. Introduction. Like the striking plumage of birds or the kaleidoscopic wings of butterflies, dragonflies and damselflies show a dazzling array of colors. Research on this order of insects serves as a conceptual framework for a number of questions about color variation, production, and function. Here, we present a summary of what is known about color production and function in adult odonates, such as its ecological importance, starting with the definition of odonate color and a description of color variation between and within species. We explain the mechanics behind the production of different types of coloration, including pigmentary absorption, structural coloration, and fluorescence. We also cover the adaptive role of color as a visual signal to members of the same or different species and its protection. We finally highlight valuable future research directions, including the need for additional biochemical, ecological, and phylogenetic analyses of odonate color. Odonate color vision. Consideration of color naturally begins with an examination of vision. Among metazoans, odonates have some of the largest eyes relative to body size, which are equipped with ca. 30,000 ommatidia each, the largest number among living insects. Shirk, 1978. Dragonflies and damselflies have some of the most advanced insect visual systems, with the ability to perceive light from almost all directions and resolve fine details in their field of view. Chauhan et al. 2014, Corbett, 1999, Land and Nielsen, 2002. Each ommatidia exhibits its own lens overlaying a crystalline cone, which is immediately above the photoreceptive cells. The ommatidia located on the dorsal surface of the eye are sensitive at between 300 and 500 nanometers wavelengths, which allows the animal to see objects clearly against the bright background of the sky. Bybee et al. 2012, Labhart and Nielsen, 1995. The ventral eye is sensitive to a wider range of wavelengths, approximately 300 and approximately 700 nanometers, facilitating. Journal of Zoology 3-1. The detection of conspecifics against different visual backgrounds. Fudahashi et al. 2015, Lancer et al. 2020. The acute zones of the eye contain larger ommatidia that permit the entrance of more photons per unit, reducing diffraction among the lenses of the ommatidia and permitting the formation of a clearer image with better color perception. Bybee et al. 2012. Specific types of odonate opsin genes produce light sensors, i.e., photosensitive intramembranous proteins, that are sensitive to different wavelengths. Bybee et al. 2012, and Chauhan et al. 2014 provided some insights on the evolution and distribution of opsons among odonats. More recently, Fudahashi et al. 2015 have identified from 15 to 33 opsin genes in odonate eyes, several of which exhibit multiple copies for color detection, including long wavelength, i.e., red, yellow, and green, short wavelength, i.e., violet and blue, and even ultraviolet sensitive opsins. The opsins are expressed differently in larvae and adults, and in ventral and dorsal regions of the eyes. Fudahashi et al. 2015. The expression variation along ontogeny is related to the versatility of morphological and ecological adaptations of these insects in the aquatic and terrestrial environments they inhabit, which implies that opsin genes may have evolved according to the habitat and/or behavior of each species. Fudahashi, 2017. Finally, odonats also exhibit optimized omatidia in their eyes that enable them to detect light polarization. Lancer et al. 2020. Adult color variation. Body color. A few days after emerging as terrestrial adults from aquatic larvae, odonats exhibit a plethora of body colors, supporting information figure S1. Before this stage, teneral imagos are pale, but this period does not last more than 10% of the adult lifespan. Corbett, 1999. The range of larval and adult odonate colors includes the entire visual spectrum, some examples shown in supporting information figure S1, as well as UV reflectance range, approximately 300 to 400 nm. Colors can be based on eye pigments such as in supporting information figure S1c, micro and nanostructures on the cuticle, i.e., structural coloration that may result in iridescent or metallic visual effects, such as in supporting information figure S1j, bright blue or whitish pruinosity derived from wax crystals, and to a lesser extent, IV fluorescence, e.g., Fudahashi, 2020, Guillermo Ferreira et al., 2014. Wing color. Most odonats have hyaline or somewhat translucent wings, yet some species have small or large wing patches formed by pigment deposition, nanostructures, and/or epicuticular wax. Examples of some types of wing color variation are shown in supporting information figure S2. Coloration can be found at the base, middle, or distal part of wings, and some species even have coloration throughout the entire wing. The coloration patterns can also vary from irregular or round patches on the wing to brown or black bands that span the wing from the costal vein to the posterior edge of the wing. Furthermore, colors such as red, brown, or black may overlap at the base of the wings. Sexual dimorphism and color polymorphisms. Color variation within a species may result in sexual dimorphism. Cooper et al. 2016, Fudahashi, 2017, Grether and Gray, 1996, with usually brighter and more conspicuous males, when sexual maturity is achieved. Corbett, 1999, Fudahashi, 2016. Hereditary color polymorphism is defined as the coexistence of at least two discrete morphs in adults of one or both sexes within a single population of a species. Huxley, 1955. Some odonate species express such color polymorphism in adults of both sexes. Fudahashi, 2017, although it is usually restricted to only one sex. Fink et al. 2005, Svensson et al. 2005, Van Gossem and Mattern, 2008. Odonata. Odonata is an order of predatory flying insects that includes the dragonflies and damselflies, as well as the Epiophlebia damsel dragonflies. The two major groups are distinguished with dragonflies, Anisoptera, usually being bulkier with large compound eyes together and wings spread up or out at rest, while damselflies, suborder Zygoptera, are usually more slender with eyes placed apart and wings folded together along body at rest. Adult odonats can land and perch, but rarely walk. All odonats have aquatic larvae called naiads or nymphs, and all of them, larvae and adults, are carnivorous and are almost entirely insectivorous, although at the larval stage they will eat anything that they can overpower, including small fish, tadpoles, and even adult newts. The adults are superb aerial hunters and their legs are specialized for catching prey in flight. Odonata in its narrow sense forms a subgroup of the broader Odonatoptera, which contains other dragonfly-like insects. Etymology and terminology. Johann Christian Fabricius coined the term odonata in 1793 from the ancient Greek odon odon, Ionic form of otis otis, tooth. One hypothesis is that it was because their maxillae are notably toothed. 1. The word dragonfly usually denotes only Anisoptera, but is sometimes used to mean all odonata. 2. Odonata enthusiasts avoid ambiguity by using the term true dragonfly. 3. Or simply Anisopteran. 4. When they mean just the Anisoptera. An alternative term warriorfly has been proposed. 5. External morphology. Size. The largest living odonate is the giant Central American helicopter damselfly Megaloprepus coriolatus, Zygoptera, pseudostigmatidae, with a wingspan of 191 mm, 7.5 in. The heaviest living odonates are Tetracanthogyna plagiata, Anisoptera, Eschnidae, with a wingspan of 165 mm, 6.5 in, and Pedalura ingentissima, Anisoptera, Pedaluridae, with a body length of 117 mm, 4.6 in, some sources 125 mm, 4.9 in, and wingspan of 160 mm, 6.3 in. The longest extant odonate is the neotropical helicopter damselfly Mecistogaster linearis, Zygoptera, pseudostigmatidae, with a body length of 135 mm, 5.3 in. 6. The smallest living dragonfly is Nanophia pygmaea, Anisoptera, Libellulidae, from East Asia, with a body length of 15 mm, 0.59 in, and a wingspan of 20 mm, 0.79 in. The smallest damselflies, and also the smallest odonats, are species of the genus Agriocnemis, Zygoptera, seen Agrionidae with a wingspan of only 17-18 mm, 0.67-0.71 in. 7. Description. Male blue ringtail. Australestes annulosus, a damselfly, Zygoptera, lestidi. Dragonfly top and damselfly bottom, wing shape and venation. These insects characteristically have large rounded heads covered mostly by well-developed, compound eyes, which provide good vision, legs that facilitate catching prey, other insects, in flight, 2 pairs of long, transparent odonata. Temporal range. Echna Unkia hovering over a pond. Scientific classification domain. Eukaryota kingdom. Animalia phylum. Arthropoda class. Insecta unranked Dicondylia subclass. Pterygoda division. Paleoptera superorder. Odonatoptera. Order. Odonata. Fabricius, 1793. Suborders. Epiprocta. Epiophlebiidae. Anisoptera, dragonflies, Zygoptera, damselflies, for extinct groups, see text wings that move independently, and elongated abdomens. They have 3 ocelli and short antennae. The mouthparts are on the underside of the head and include simple chewing mandibles in the adult. 8. Flight in the odonata is direct, with flight muscles attaching directly to the wings, rather than indirect, with flight muscles attaching to the thorax, as is found in the Neoptera. This allows active control of the amplitude, frequency, angle of attack, camber and twist of each of the 4 wings entirely independently. 9. In most families, there is a structure on the leading edge near the tip of the wing called the pterostigma. This is a thickened, hemolymph-filled and often colorful area bounded by veins. The functions of the pterostigma are not fully known, but it most probably has an aerodynamic effect and may also have a visual function. More mass at the end of the wing may also reduce the energy needed to move the wings up and down. The right combination of wing stiffness and wing mass could reduce the energy consumption of flying. A pterostigma is also found among other insects, such as bees. 10. The nymphs have stockier, shorter, bodies than the adults. In addition to lacking wings, their eyes are smaller, their antennae longer, and their heads are less mobile than in the adult. Their mouthparts are modified, with the labium being adapted into a unique prehensile organ called a labial mask for grasping prey. 11. Damselfly nymphs breathe through external gills on the abdomen, while dragonfly nymphs respire through an organ in their rectum. 8. Evolution. Fossil history. The giant upper Carboniferous dragonfly ancestor, Meganora mani, had a wingspan of about 680 mm, 27 in 12. Members of the crown group Odonata first appeared during the late Triassic, 13th though members of their total group, Odonatoptera, first appeared in the late Carboniferous, making them one of the earliest groups of winged insects. 13. The fossils of odonats and their cousins, including Paleozoic, giant dragonflies, like Meganeropsis permiana from the Permian of North America, reached wingspans of up to 71 cm, 28 in, and a body length of 43 cm, 17 in, making it the largest insect of all time. 14. This insect belonged to the order Meganesoptera, the griffinflies, related to odonats but not part of the modern order Odonata in the restricted sense. They have one of the most complete fossil records going back 319 million years. 15. The Odonata is closely related to mayflies and several extinct orders in a group called the Paleoptera, but this grouping might be paraphyletic. What they do share with mayflies is the nature of how the wings are articulated and held in rest. 16. Tarsiphlebiidae is a prehistoric family of Odonatoptera that can be considered either a basal lineage of Odonata or their immediate sister taxon. 17. Phylogeny. The phylogenetic tree of the orders and suborders of odonats according to Bybee et al. 2021. 18. Odonata. Zygoptera, damselflies. Epiprocta. Anisozygoptera. Anisoptera, dragonflies. Taxonomy. In some treatments, 19, the Odonata are understood in an expanded sense, essentially synonymous with the superorder Odonatoptera, but not including the prehistoric Protodonata. In this approach, instead of Odonatoptera, the term Odonatoidea is used. The systematics of the Paleoptera are by no means resolved. What can be said, however, is that regardless of whether they are called Odonatoidea or Odonatoptera, the Odonata and their extinct relatives do form a clade. 20. The Anisoptera was long treated as a suborder, with a third suborder, the Anisozygoptera, ancient dragonflies. However, the combined suborder Epiprocta, in which Anisoptera is an infraorder, was proposed when it was thought that the Anisozygoptera was paraphyletic, composed of mostly extinct offshoots of dragonfly evolution. The 4 living species placed in that group are, in this treatment, in the infraorder Epiophlebioptera, whereas the fossil taxa that were formerly there are now dispersed about the Odonatoptera, or Odonata sensu lato. 21-22 World Odonata List considers Anisoptera as a suborder along with Zygoptera and Anisozygoptera as well understood and widely preferred terms. The 4 living Ovipositing flight of 2 pairs of azure damselflies, Cinegri and Puella. Odonats are aquatic or semi-aquatic as juveniles. Thus, adults are most often seen near bodies of water and are frequently described as aquatic insects. However, many species range far from water. They are carnivorous, or more specifically insectivorous, throughout their life, mostly feeding on smaller insects. 27. Ecology and life cycle. Male Odonata have complex genitalia, different from those found in other insects. These include grasping cerci at the tip of the abdomen for holding the female, and a secondary set of copulatory organs located between the second and third abdominal segment in which the spermatozoa are stored after being produced by the primary genitals, whose external opening is known as the genital pore, on the ninth abdominal segment. This process is called intra-male sperm translocation, ST. 28-29. Because the male copulatory organ has evolved independently from that in other insects, it has been suggested the stem group dragonflies had external sperm transfer. 30. To mate, the male claspers grasps the female by the thorax, zygoptera, or head, anisoptera, while the female bends her abdomen so that her own genitalia can be grasped by the copulatory organ holding the sperm. This is known as the wheel position. 8. In anisoptera, males often mate while flying, lifting the females in the air, which typically lasts from a couple of seconds to a minute or two, whereas the males in zygoptera mate while perched. They might even move to different spots during the mating process, which can make it last longer, anywhere between 5 to 10 minutes. Male Odonata are very competitive when it comes to mating that in some species, the males use the cerci located at the tip of the abdomen to remove the sperm of a rival males from the female and put in his own. 31-32. Eggs are laid in water or on vegetation near water or wet places, and hatch to produce pronymphs which live off the nutrients that were in the egg. They then develop into instars with approximately 9 to 14 molts that are, in most species, voracious predators on other aquatic organisms, including small fishes. The nymphs grow and molt, usually in dusk or dawn, into the flying teneral immature adults, whose color is not yet developed. These transform into reproductive adults. Odonats can act as bioindicators of water quality in rivers because they rely on high-quality water for proper development in early life. Since their diet consists entirely of insects, odonat density is directly proportional to the population of prey, and their abundance indicates the abundance of prey in the examined ecosystem. 33. Species richness of vascular plants has also been positively correlated with the species richness of dragonflies in a given habitat. This means that in a location such as a lake, if one finds a wide variety of odonats, then a similarly wide variety of plants should also be present. This correlation is not common to all bioindicators, as some may act as indicators for a different environmental factor, such as the pool frog acting as a bioindicator of water quality due to its high quantity of time spent in and around water. 34. In addition, odonats are very sensitive to changes to average temperature. Many species have moved to higher elevations and latitudes as global temperature rises and habitats dry out. Changes to the life cycle have been recorded with increased development of the instar stages and smaller adult body size as the average temperature increases. As the territory of many species starts to overlap, the rate hybridization of species that normally do not come in contact is increasing. If global climate change continues, many members of Odonata will start to disappear. Because odonats are such an old order and have such a complete fossil record, they are an ideal species to study insect evolution and adaptation. For example, they are one of the first insects to develop flight, and it is likely that this trait only evolved once in insects. Looking at how flight works in odonats, the rest of flight can be mapped out. 5. Dragonflies and damselflies are predaceous both as immatures and adults. The adults are quick, agile flyers that are generally considered beneficial because they feed on large numbers of small, flying insects like gnats and mosquitoes. Legs are used either as a basket for catching prey or as grapples for clinging to emergent vegetation. Eggs are laid singly in fresh water. Females often hover over open water and dip their abdomen as they oviposit. Eggs hatch into aquatic immatures, naiads, that feed opportunistically on other forms of aquatic life including mayfly naiads, small crustaceans, annelids, and mollusks. Some of the large dragonfly naiads will even attack small fish and tadpoles. All immature Odonata have a specialized labium for catching prey. Folded under the head and thorax when not in use, the labium can be extended rapidly toward potential prey. Hook lobes at the tip of the labium grasp or impale the prey and draw it back to the mouth as the labium retracts. Damselfly naiads are usually more slender than dragonfly naiads and have 3 leaf-like gills at the end of the abdomen. Dragonfly gills are located internally, within the rectum, where bellows-like contractions of the rectal muscles cause oxygenated water to circulate in and out. Appearance of dragonflies. Immatures. 1. Labial, mask, adapted for catching prey. 2. Body robust. Adults. 1. Antennae short and bristle-like. 2. Compound eyes large, often covering most of the head. 3. 4 membranous wings with many veins and crossveins. 4. Base of hind wing broader than forewing. 5. 1 distinctively pigmented cell, stigma, on leading edge of wing. 6. Abdomen, long and slender. Appearance of damselflies. Immatures. 1. Labial, mask, adapted for catching prey. 2. 3 leaf-like gills at rear of abdomen. 3. Body usually long and slender. Economic importance. Most dragonflies and damselflies are regarded as beneficial insects because they feed on small flying insects such as mosquitoes. They may also catch and eat honey bees, then they are regarded as pests by the beekeepers. In some parts of Europe, dragonflies are considered a threat to the poultry industry because they transmit Prosthogonomus pellucidus, a parasitic flatworm. Dragonfly naiads become infected by ingesting cysts of the flatworm. These cysts survive into adulthood of the dragonfly and may spread to birds, particularly poultry, that catch and eat the adult dragonflies. The flatworm cysts dissolve in the bird's intestine and infection spreads into the cloaca and reproductive organs. The Dutch have a maxim: hide the hens, the dragonflies are coming. Major families. Dragonflies. Echinidae darners. These insects are notable for their large size and brilliant blue or green coloration. Includes the common green darner, Anax junius. Libellulidae, common skimmers. This is the largest family in the order. It contains many species with dark spots on the wings. Gomphidae clubtails. These dragonflies have the terminal abdominal segment swollen, hence the common name. Adults. 1. Antennae short and bristle-like. 2. Compound eyes large, often covering most of the head. 3. 4 membranous wings with many veins and crossveins. 4. Base of wings narrow, stalk-like. 5. 1 distinctively pigmented cell, stigma, on leading edge of wing. 6. Abdomen, long and slender. Calopterygidae, broad-winged damselflies. The wings of these insects are shaped like the seeds of a maple tree. Cynogryonidae, narrow-winged damselflies, small, delicate insects. The body is usually black with blue markings. At rest, the wings are held together over the back. Lestidae, spread-winged damselflies. These damselflies rest with the body nearly vertical and the wings partly outspread. Fun facts. The compound eyes of some dragonflies may have up to 28,000 facets. Some naiads can shoot out their labium and catch prey in only 25 milliseconds. Scientists have documented large-scale migrations of dragonflies. One swarm was observed 1,400 kilometers off the coast of Australia. Some immature damselflies establish feeding territories, areas that are defended against invasion by other conspecifics. Territorial species develop more rapidly and produce larger adults than other non-territorial species. Many adult male dragonflies establish and defend territories along the perimeter of a lake or stream. Females will mate only with males that hold a territory, so population density is somewhat regulated by territory size. Male Odonata have claspers at the end of their abdomen, but no external genitalia. Before finding a mate, a male attaches a spermatophore to his second abdominal segment. He then grabs a female around the neck with his claspers and she retrieves the spermatophore with the genital opening of her abdomen. Most dragonfly naiads can move forward by jet propulsion. Rapid contraction of the rectal muscles forces water out the rear end and shoots the insect forward. Male damselflies, and perhaps some dragonflies, have a special flagellum associated with the copulatory organ that can reach into a female's body and remove sperm deposited by another male in a previous mating. Dragonflies are known by many interesting common names, including snake doctors, devil's darning needles, and mosquito hawks. Bull. 60, 353 to 300. The odonate mating system. Communication, and sexual selection. If two species are to maintain their genetic integrity and symmetry, devices must exist which prevent interspecific gene flow. These devices have been called reproductive isolating mechanisms and might operate before or after mating. Dobzhansky, 1937. Two contrasting models describe the origin of reproductive isolating mechanisms. The Darwin-Muller-Meyer theory, cf. Meyer, 1988, claims that premating isolating mechanisms arise as byproducts of natural selection in allopatry. They are thus no direct result of selection for reproductive isolation. According to the Wallace-Dobzhansky model, cf. Meyer, 1988, reproductive isolating mechanisms evolve as a direct response to natural selection against hybridization to prevent gametic wastage. Species isolation is closely related to mate recognition and sexual selection. Canachero, 1980, Canachero and Boak, 1987, Varale, 1988. Particularly behavioral biologists, e.g., West Eberhard, 1983, 1984, Holdobler, 1984, Thornhill and Alcock, 1984, Eberhard, 1985, have revived the awareness that sexual selection plays a major role in the evolution of sexual signaling systems, i.e., of mate recognition. The odonate mating system has already been described and classified on several occasions, e.g., Corbett, 1962, 1980, Thornhill and Alcock, 1983, Alcock, 1984, Wahage, 1984, Conrad and Pritchard, 1992. Isolating mechanisms have been the subject of several discussions in odonate biology. Johnson, 1962, Watson, 1966, Paulson, 1974, Robertson and Patterson, 1982, Tennyson, 1982. Despite this body of information, my goal here is to propose another review in order to elucidate the relation between sexual selection and sexual signaling systems in odonates. I thus begin by describing communication systems in the odonate mating process. Then I develop the idea that sexual selection operates as a major evolutionary force in odonate sexual communication devices, which are subsequently discussed as potential isolating mechanisms. Communication and sexual selection. Using Emlyn and Oring's 1977 scheme for an ecological classification of animal mating systems, Conrad and Pritchard 1992 investigated the relative influence of intront intersexual selection on the evolution of the odonate mating system. They partitioned the odonate mating system into non-resource and resource-based systems, which they subdivided into free female choice, female-control, and encounter-limited mating, and into resource control and resource limitation, respectively. Intrasexual selection predominates in female control and resource control systems, whereas intersexual selection is prevalent in the free female choice system. Both intront intersexual selection shape the resource limitation system. Conrad and Pritchard, 1992. 354 T.J. Batten. Characters acted on by sexual selection are behavioral and morphological traits involved in sexual communication, e.g., Holdobler, 1984, West Eberhard, 1984. Intrasexual selection mainly targets communication devices involved in male interactions. Intersexual selection arises through preferences for a particular trait in the other sex. Female mate choice is now widely recognized as one of the primary mechanisms of intersexual selection, CEG, Majerus, 1986, Kirkpatrick and Ryan, 1991. Females might discriminate among males on the basis of visual, auditory or olfactory cues, but mainly, focus on the resources males are offering. In species in which females choose males on the basis of resources and where males compete for these resources, intersexual selection, i.e., female choice might reinforce intrasexual selection, i.e., male-male competition, Searcy, 1982. Interrenal competition. In resource defense polygyny, males defend resource patches attractive to females and enhance thereby their reproductive success, Emlyn and Oring, 1977. The restriction of resources and their uneven distribution in time and space produce territorial patches of different quality and thus induce intermale territorial competition. Odonate territoriality is outstanding among insects, see Baker, 1983, Fitzpatrick and Wellington, 1983. The key resource in odonate territories is the potential oviposition site, e.g., Wahage, 1973, Thornhill and Alcock, 1983, Alcock, 1990, Meek and Herman, 1990, Conrad and Pritchard, 1992. The implication of other resources, e.g., feeding sites, in mating success should not, however, be played down, since life history is reflected in the fighting ability of contestants as well. Ubukata, 1987, postulates that odonates are likely to assess the patch quality by the abundance of oviposition sites and by cues such as feeding or female arrival rates. Escalated territorial contests can be very costly and energy reserves are crucial to reproductive success, see Martin and Wahage, 1990. Therefore, in order to reduce such costs and to enhance thereby reproductive success, males rely heavily on appropriate communication systems. Using Alcock's 1984 criteria of communication channels, it is the visual channel which best performs this function. In fact, its transmission rate and fade-out time are fast, the sender locatability is easy, and the costs to the sender are low to moderate, see Alcock, 1984. It is exactly the visual communication channel which predominates in odonate intermale competition. Patterns of intermale competition exhibit high diversity, ranging from simple threat display to ascending chase flight. For example, see the numerous citations in Corbett, 1962, 1980, Wahage, 1984, Ubukata, 1987. Classic examples of signaling devices in intermale competition are wing coloration patterns in Calopteryx, CEG, Wahage, 1973, 1988, and in Libellula, Moore, 1990. Frantsevich and Mokrushov, 1984, experimentally showed that the wing spot of Sympatrum males releases visual response in conspecific males. Territorial males with specific wing patterns in fact recognized conspecific intruders more efficiently than species without a wing spot did. In Platycypha species, male-male interactions involve display through tibial vibration, Robertson, 1982. In libellulid species, visual signals are also emitted by abdomen coloration, Jacobs, 1955, Van Buskirk, 1987. Courtship. A prime example of an epigamic trait is undoubtedly the wing spot in Calopteryx species. It constitutes the basic visual signal in both sexes which, depending on the way it is displayed, transmits different information. Courtship patterns involving the wing spot as the chief communication device are, for instance, the male cross display, see Wahage, 1973, 1988, or courtship arc, see Meek and Herman, 1990. Unreceptive females reply to courting males through the refusal display, see Wahage, 1979, 1984, i.e., by spreading all four wings and raising the abdomen. Another particularly dramatic example of epigamic traits is known from Platycypha caligata. Here the male courts by hovering around the female and by displaying his vibrating flattened tibi, Robertson, 1982. In damselflies, the courtship repertoire also includes wing vibration, variation of the posture angle of the body, or downwards flexing of the abdominal tip, see review by Utsuri, 1988, for more details. At this stage of the copulation process, mechanisms of precopulatory female choice might firstly act, cf. Partridge and Holliday, 1982, thus leading to tandem linkage or not. Meek and Herman, 1990, recently showed that choosy females of Calopteryx species rejected 70–88% of male cross displays, whereas the subsequent mating stage courtship arc generally encompassed less discriminatory power and subsequently led to tandem formation. Tandem linkage. This important mating stage is characterized by the shift of the communication channel from visual to tactile modalities. Genitalic structures involved in tandem formation are the male anal appendages, the female occipital region in most Anisopteran, and the prothoracic and/or metathoracic complex in Zygopteran species. Communication through tactile stimuli was first evidenced by Krieger and Krieger-Leubel, 1958, and Leubel, 1958. Females of the Zygopteran genera Lechtes and Ischnura refused to copulate with males having experimentally altered anal appendages. Using a similar experimental approach, Robertson and Patterson, 1982, corroborated the tactile stimuli hypothesis. Near Eastern Cynegreonid females were shown in 1993 to have their mesostigmal complex developed into tactile recognition sites, see Figs 2-8, similar to those originally described for Enolagma, Robertson and Patterson, 1982. Jerzitsa, 1974. 1975 provides additional evidence from ultrastructural analyses. Odinate mating system 355. Thus far no clearly defined concept of communication through tandem linkage has been advanced. Tennyson, 1982, merely suggests that mechanical communication, in the sense of mechanical isolation, has been misinterpreted and actually involves tactile stimuli. Data from Enolagma, Robertson and Patterson, 1982, and Cynegreon Figs 2-8 suggest, however, that both mechanical and tactile stimuli should be regarded as two distinct components in the communication system. In fact, mechanoreceptors are non-randomly scattered on the female mesostigmal plate, Figs 2-5. They are restricted to exactly those sites directly involved in male contact and actually making up the species specificity of the plates. Female tactile components thus receive the basic signal, which is modulated by the grade of the mechanical fit between male and female structural components. When structural incompatibility exceeds a critical threshold, tactile stimuli are triggered off in the thoracic complex and thus imply female assessment of whether to continue or abandon the mating sequence. This concept matches Eberhard's 1985 hypothesis fairly well, which applies female choice to animal genitalia. Eberhard supposes that male genitalia assume internal courtship, which is assessed by the female on the basis of mechanical and stimulatory cues. There is, however, no direct experimental evidence on female choice during this stage. The only supporting argument might be deduced from the key position coming up to the female during this mating stage. By bending her abdomen forward or not, it is notably the female who determines whether genitalic coupling takes place or not. Genitalic coupling and sperm transfer. Genitalic coupling is achieved through the interaction of the secondary structures, e.g., hamules, plates, of the male copulatory complex and the female gonapophyses and/or genital plates. Seafow, 1970, Miller, 1981, 1984, or Miller and Miller, 1981, for a comprehensive account on the functional morphology of secondary genitalia. Many authors have argued that discrimination, male and/or female acceptance or rejection, occurs during genitalic coupling, e.g., references in Eberhard, 1985, Watson, 1966, which therefore implies intersexual communication. However, I am not aware of any sound evidence for tactile communication through these structures. The penis itself is not involved in genitalic holding, but is obviously designed as an instrument for sperm competition, see Wage, 1979, 1984, 1986. The most extravagant morphological traits shaped for the dual function of sperm removal and transfer are flagella and spines are similar microstructures on the penis surface, see Wage, 1986, Miller, 1990, 1991. Campaniform cincilla in the vagina of libellulids, see Shiva Jati, 1987, suggests that tactile communication occurs in this area. Whether these receptors imply intersexual communication remains speculative. Differentiation in penis armature of 3 Colopterygid species might hint, however, at intersexual communication through penis stimulation, cf. Eberhard, 1985, Adams and Herman, 1991. By contrast, Shiva Jati's 1987 and Miller's 1991 assumption that campaniform cincilla might be excited by the presence of eggs in the duct, and that their activity evokes sperm release, sounds more plausible. Sperm competition is now regarded as a crucial mechanism of postcopulatory intrasexual selection. It manifests perhaps its most dramatic form in Odonata, and its research probably ranks as the most advanced in this order. Burkhead and Hunter, 1990. Conspecific males compete to fertilize the eggs in a female by removing the sperm previously stored by a rival and by placing their own sperm into the female storage organ. Sperm competition is fairly well documented in 3 Zygopteran families: Colopterygidae, Wage, 1979, Cynegreonidae, e.g., Miller and Miller, 1981, Fink, 1984, Wage, 1984, and Lestidae, Wage, 1982, and in the Anisopteran family Libellulidae, Shiva Jati, 1984, Miller, 1991. Prerequisites for sperm competition include sperm storage, delayed fertilization, and multiple copulations by females. Females may gain increased genetic heterogeneity of their offspring from multiple mating, thus resulting in increased female fitness, see Lohman et al., 1988. Moreover, sperm competition may supply females with superior mates or guarding, but can also be associated with costs. Costs arise from time and energy investments, but chiefly from the reduction of the genetic heterogeneity in offspring, Knowlton and Greenwell, 1984. Sperm competition thus promotes a sexual conflict between female interests and the male paternity assurance. Oviposition. After copulation, females usually oviposit promptly, with or without guarding. Guarding behavior can be divided into a contact guarding, the male remaining attached to the female through tandem linkage, and b non-contact guarding, the male hovering or perching near the female. Wage, 1984. Utsuri, 1989, recently produced some suggestive evidence for information flow from the female to the male during contact-guarded oviposition, i.e., tandem linkage. Postcopulatory mate guarding during oviposition is the prevalent adaptation in odonats to prevent sperm competition from other males, Waj, 1984. It reduces the takeover of females by unpaired males, and thus assures the paternity of the male's present mate, i.e., the egg batch fertilized by his stored sperm. Moreover, interference from non-pairing males might cause costs to the female. Therefore, postcopulatory guarding is an important interference-avoiding mechanism in odonats. The capacity for avoiding interference is correlated with the male's dominance and can thus be subjected to female assessment through morphological and/or behavioral traits, cf. Searcy, 1982. 356 T.J. Batten. Furthermore, an essential point of female choice centers on the oviposition site itself, notably on patch size, oviposition substrate, location, presence of other females or disturbance, see Alcock, 1987, Waj, 1987, Meek and Herman, 1991. Females enhance their reproductive success by choosing those males offering an oviposition patch likely to guarantee survival and successful development of their offspring. Females can reject the oviposition patch selected by their partner prior to CEG, Robertson, 1982 and references therein, or after copulation, Alcock, 1990. Rejecting the oviposition patch only after copulation, females can solicit additional copulations and thus foster sperm competition by males of neighboring territories. Species specificity and mate recognition. Beside the effect of shaping mating systems, Emlyn and Oring, 1977, Conrad and Pritchard, 1992, sexual selection is known to act on behavioral and morphological traits as well. It should, however, be stressed that the exact effects of the different selective mechanisms are not yet detected. Moore, 1990, for instance, was able to relate natural variation in the male wing coloration and body size to mating success in a libellulid dragonfly. Both traits represent important fitness components in the species under study. Moreover, Fink, 1992, has shown that sexual selection favors larger males of a neotropical damselfly in territorial contests, thus resulting in higher mating efficiency relative to smaller males. Eberhard, 1985, developed the idea that rapid and divergent evolution of sex-involved morphologies is generated by sexual selection. Alternative hypotheses, amongst which the most venerable probably is the lock and key hypothesis, see Shapiro and Portner, 1989, for a critical review, or reviewed in Eberhard, 1985. Land, 1981, 1982, demonstrated in quantitative genetic models of Fisher's runaway process, initially envisioned by Fisher, 1930, for review c. e.g., Kirkpatrick and Ryan, 1991, and Maynard Smith, 1991, that female choice alone can lead to speciation even in the presence of moderate gene flow, i.e., non-geographic isolation speciation. Thus, if a female mating preference system is genetically unstable, even slight geographic variation in natural selection of males can initiate runaway sexual selection. Evolution of secondary sexual characters will of course be facilitated in allopatric speciation. In the allopatric speciation model, gene flow is interrupted by an external geographic ecological barrier which divides the ancestral gene pool into two subunits. During this phase of the speciation process, intrinsic isolation mechanisms might evolve in both subunits, most likely as pleiotropic consequences of other evolutionary shifts. Bach, 1986. If the intrinsic isolating mechanisms are not effective, gene flow will produce one interbreeding unit after secondary contact. If they are effective, however, both populations will become sympatric, thus implying reproductive interactions. As genetic isolation must not necessarily involve reproductive isolation, individuals of both incipient species, although isolated through intrinsic mechanisms, might copulate. Such reproductive interactions, however, yield offspring of reduced viability, thus being nothing but gametic wastage. To avoid these considerable reproductive costs, rapid divergence of sexual signaling systems is favored. Bach, 1986, and might even be reinforced by mechanisms of Fisher's runaway sexual selection. Here, sexual signals develop reproductive character displacement, see Butlin, 1987, thus decreasing genetic exchange between both incipient species, and therefore functioning in prezygotic isolation. Odonats might potentially discriminate amongst conspecific and heterospecifics in a multi-stage process, Fig. 1, which covers both sequentially and physiologically different mating stages. The visual stage is undoubtedly important in species with elaborate behavioral displays. Reproductive character displacement at this discrimi. Intrasexual selection, intersexual. 9. / Mating sequence. Male-male encounter. T-male-male competition. I. Tau male-female encounter courtship. Display. Tandem linkage. Asterisk. GENITALIC coupling oviposition. Information flow. E. G. Wing, abdomen receiver. Iota. Omicron I. E. G. Wing. 2 abdomen zeta 1 Saint discrimination. Stage. I1. And 1 appendages 1. Plus J2 ill discrimination thoracic degree. Complex eta. O. And zeta appendages RR. Thoracicum complex gamma. Figure 1 conceptual model of the fundamental odonate mating system and the communication therein involved. Each vertical arrow links a series of stages in the reproductive process. To the right of the mating pathway, the sender and receiver are given for each communication channel of each mating stage. Where no sound evidence for communication was available, question marks indicate that lack. For details, see text. Odonid mating system 357. Figs 2 to 8 structural components involved in copulation in Near Eastern Cynegrian species. Female mesostigmal plates are species-specific in all 4 taxa: C. syriacum, 2, C. ponticum, 3, C. puella, 4, C. intermedium, 5. Mechanoreceptors, 6, similar to those described from Enilagma subfurcatum, C. Robertson and Patterson, 1982, are confined to those areas directly contacted by the male appendages, cf. Bullard, 1981, for functional morphology in Cynegrian cytulum. Mechanoreceptors, 7 and 8, on the carinal complex of female C. puella. Nation state. Conclusion. Most of the points outlined here have already been discussed separately before, as indicated by the references. However, their connection with the speciation process emphasizes the intimate relation between communication, sexual selection and mate recognition. Whether considered in the context of the biological species concept, see Hauser, 1987, for review, or in the context of the recognition concept Patterson, e.g. 1985, the role of mate recognition systems remains undisputed in promoting species integrity. The biological concept views mate recognition as a species isolating function. By contrast, Patterson, 1985, postulated that the specific mate recognition system, SMRS, shared by one species has evolved to promote effective syngamy between individuals of that species. The components of the Odonid mate recognition system, of which only 2 are reliably known so far, are well matched with Patterson's SMRs. As Varale, 1988, rightly pointed out, the recogneltion concept, as currently in use, denies the mechanisms of sexual selection as an evolutionary force. This is a major deficiency of the model as it limits its applicability. Both the biological and the recognition concept emphasize strong stabilizing selection on sexual signaling systems and therefore lessen intraspecific variation as an important target for sexual selection. Ryan, 1990. Considerable efforts have already been made in Odonid sexual biology towards the solution of the intriguing puzzle of evolution. It follows most obviously from this review that interest in empirical tests is badly needed. Future interest should thus focus on the research avenue of sensory biology for a better understanding of female choice. Moreover, phylogenetic analyses centering on sexual signaling systems should be strengthened. Odonids may thus become a leading vehicle for testing the applicability of current species concepts. Our understanding of what a species actually is in Odonids will certainly benefit from these approaches. Studies on the reproductive behavior of dragonfly, Pantala flavescens fabricius, 1798 Odonata, insecta, arthropoda, in Aravalli Range and desert ecosystem of Rajasthan, India. For 270 million years, dragonflies with their 4 long independent membranous wings and long bodies have remained unchanged in their essential form. Dragonflies were the first creatures to truly command the air of this earth. They are the fighter planes of the insect world, hovering easily, flying fast, and maneuvering seemingly instantly. They are flying dragons only to flies and mosquitoes, however, they cannot bite or sting us. They are amphibious hemimatable insects having the aquatic egg and larval nymph stages, while the adults are terrestrial. Dragonflies possess well-diversified behavioral patterns, including larval and adults feeding behavior, emergence pattern, post-emergence dispersal and the reproductive behavior, of which, the last one is the most significant. Though, the generalized patterns of these behaviors are quite same among the world of dragonflies, till detailed study reveals that, they vary greatly from species to species. Hence, in this paper an attempt has been made to provide the detailed information on reproductive behavior of dragonfly, Pantala flavescens fabricius. The different aspects of reproductive behaviors on different species of dragonflies studied throughout the world by various workers such as Williamson, 1906, Williams, 1936, Hodgkin and Watson, 1958, Erickson, 1960, Ito, 1960, Moore, 1960, Johnson, 1961, 1962A and B. Corbett, 1962, Pajunin, 1962, 1963, Kyuda, 1964, Bick and Hornuff, 1966, Bick and Sulbach, 1966, Higashi, 1969, Waj, 1979, Arai. Bio Bulletin 3, 1, 60. 1972, Bic 19 19908. Materials and methods. The reproductive behavior of Pantala flavescens fabricius was studied in different localities of Aravalli Range, i.e. Jodhpur, Mount Abu and Udaipur, and desert ecosystem, i.e. Bikaner, Barmer and Jaisalmer, of Rajasthan during April 2008 to March 2015, Figure 1. The observation on reproductive behaviors of Pantala flavescens has been conducted in the field from morning 7 a.m. to evening 6 p.m. for about 52 days in the different localities in the months of June to October. During study period and observation were also recorded by taking photographs in the field by using Nikon SLR D70, D90, D7000 cameras with close-up and telelens 80 to 400 mm attachments. Results and discussion. The Odonata exhibit two periods during their entire life, i.e. the pre-reproductive period and the reproductive period. The pre-reproductive period is the period in between emergence and reproduction. The main behavior of a dragonfly during this period is dispersal after emergence and feeding to help the teneral adults in becoming fully mature adults within a few days. Hence, the pre-reproductive period is also called maturation period. It does not mean that in the reproductive period, the dragonfly remains on fast. But together with the feeding behavior, the most spectacular phenomenon of this period are the different types of reproductive behavior, which comprises territoriality, pre-copulatory tandem or before-wheel tandem position, copulatory wheel formation, post-copulatory tandem or after-wheel tandem position and oviposition. The reproductive behavior of Odonata comprises adult behavior, which leads to successful mating and oviposition. As a result of southwestern monsoon, the swarms of adults of Pantala flavescens, Fabricius, appeared in June in the study area and disappeared with the decline of monsoon in November. The 52 different observations on the reproductive behavior of Pantala flavescens, Fabricius, has been studied during 2008-15, out of which 24 cases happened without interference. Figure 1, showing the area Rajasthan state, desert ecosystem and Aravalli range. Figure 2. Figure 2. The different aspects of reproductive behavior of Pantala flavescens, Fabricius, studied in the field were as given below. A. Territoriality. The males showed a strong territorial behavior. The activity and appearance of males and females at the reservoir site depended on the sunlight. Generally the males arrive at the rendezvous during 7 o'clock to 8:30 a.m. The females appear from the surrounding vegetation late at about 9 o'clock to 10:30 a.m. Males, after arrival, choose a base perch on the stem of vegetations or on some dry plants, stones, etc. No particular preference for perch was observed. The perch formed the center of a circular territory with a radius of about 4 to 6 meters, which was defended by the resident male from the inter- and intraspecific invasions. B. Before-wheel tandem. When the female entered into the territory of a resident male, the later tried to catch her prothorax by its anal appendages. Then the male in air caught the female and formed the tandem. The before-wheel tandem lasted for 10 to 40 seconds in the air. C. Courtship and wheel formation. After this, the pair in tandem formed the wheel position in the air, in which the vulvar region of the female get interlocked with the secondary copulatory apparatus of the male. The duration of the wheel position varied from 70 seconds to 3 minutes. Generally, the formation of wheel does not need any support and occurs during flight, and also the pair in courtship remains in the air without any perching till end. However, in some cases supports was used as a hiding place to avoid interference by other males. Intramale sperm translocation was not witnessed. However, in some cases supports was used as a hiding place to avoid interference by other males. Intramale sperm translocation was not witnessed before wheel position and hence can be predicted to occur before the males arrive at the rendezvous. D. After wheel tandem. Just after breaking of the wheel, the male lowered the female and in tandem took flight in air. The after wheel tandem lasts for 20 to 30 seconds. E. Oviposition. After this, the female in tandem oviposit, moving repeatedly forward and backward along a straight line in less dense areas without interference towards the water surface or sometime by mistake on a shinning surface of boundary walls of reservoir, on roads and on vehicles. The males in tandem aggressively guarded their females during oviposition. The females after oviposition took some rest hiding at the surrounding vegetation. Females generally take 60 seconds to 2 minutes for completing the oviposition without interference. The total duration of reproductive behavior of Pantala flavescens, Fabricius, lasts for 3 to 6 minutes. In both damselflies and dragonflies, the rendezvous is at or near the oviposition site. In every case, mature males arrive at the rendezvous earlier in the season and in the day than females, and form the territory before the females arrive, which agree with the observation of Corbett, 1980. Williamson, 1906, noted the reproductive behavior in some zygopterans. Williams, 1936, observed the oviposition of Megalogrion oahuense. Moore, 1952a and b, described the oviposition of Sympatrum striolatum as well as the territoriality in some dragonflies. Hodgkin and Watson, 1958, recorded the breeding of some dragonflies in temperate water. Erickson, 1960, and Ito, 1960, worked on the oviposition of Enalagma exulsans and territoriality of Orthetrum albostylum speciosum respectively, while Moore, 1960, again described the behavior of adult dragonflies in detail. The breeding behavior and oviposition of Heterina americana, H. titia, Colopteryx maculatum and Pachydiplax longipennis were studied by Johnson, 1961, 1962a and b, respectively. Corbett, 1962, in his book, A Biology of Dragonflies, gave an account on the reproductive behavior of different dragonflies. Bio Bulletin, 2017, Volume 3, 1, 67-73, Sharma 70. From different parts of the world. Pajunan, 1963, worked out the reproductive behavior of Leucorinia dubia and L. rebecunda as well as the influence of population density on the territorial behavior of the later one. Chiuta, 1964, made some important field observations on the adult behavior of Leucorinia pectoralis. Bick and Hornuff, 1966, studied the reproductive behavior of Enalagma aspersum and E. exulsans, while Heterina americana was dealt by Bick and Sulbach, 1966. Higashi, 1969, observed the territoriality of Crocothemis cervilia. Wahage, 1979, worked on some aspects of the reproductive behavior and ecology of Calopteryx maculata, and also studied its reproductive isolation from C. aquabilis. Reproductive behaviors of Orthetrum albostylum speciosum and O. triangulare melania were studied by Arai, 1972. Bick, 1972, gave a review of the territorial and reproductive behavior in Zygopteran, whereas the substrate utilization during reproduction by Argia plana and Amoesta was studied by Bick and Bick, 1972. Furtado, 1972, highlighted the reproductive ethobiology of Ischnera senegalensis and Pseudogrian microcephalum, while Hamer, 1972 and 1973, made exhaustive studies on the reproductive behavior of the genus Calopteryx in Palearctic region. Consiglio, 1974, studied the behavior of reproduction of Platycypes caligata, while Copera marganipes and C. vittata acudimargo were dealt by Furtado, 1974. Green, 1974, observed territoriality of some Nigerian dragonflies. Jerzitsa, 1974, Mizuta, 1974, Parr and Parr, 1974, Sakagami et al. 1974 also worked on different aspects of the reproductive ethobiology of dragonflies. Arai, 1975, studied the adult behavioral patterns of Onychogomphus veridicostis. Gossam, 1999, studied the male choice for female color morphs in Ischnera elegans. Gossam et al. 2001, studied the frequency-dependent malamate harassment and intraspecific variations in its avoidance by females of Ischnera elegans. Cordero, 2000, made an analysis of multivariate selection in a non-territorial damselfly. Schultz et al. 2001, studied pursuit of heterospecific targets by territorial amberwing dragonfly Parathemis tenera. Switzer, 2002, studied the individual variations in the duration of territory occupation by males of Parathemis tenera. Cordola Angular et al. 2003, studied the sperm competition in Odonata, the evolution of female sperm storage and ribosperm displacement. Kumar and Prasad, 1977, made an excellent study on the different aspects of reproductive behavior of Neurobasis chinensis. Begum et al. 1982, commented on the sexual behavior and oviposition of Zizoma petiolatum. Prasad and Ghosh, 1982, recorded the reproductive behavior of Urothemis signata. Studious in the reproductive behavior of Ischnera A. Aurora, Seriagrian coromandelianum and Chloronera quadrimaculata were carried out by Srivastava and Babu, 1984, 1985a and b. Prasad, 1986, worked on the reproductive behavior of Thylaemus tilarga, whereas Mitra, 1987, made some comments on its breeding habitats. Begum et al. 1990, shed light on the reproductive behavior of Rhodothemis rufa. Prasad, 1990, 1991, made some important observations on the reproductive behavior of Seriagrian coromandelianum, Pseudogrion rubriceps and Brachythemis contaminata. Chowdhury and Karim, 1994, observed the reproductive behavior of Copera annulata, while Srivastava et al. 1994 made a detailed study on the behavior of reproduction and oviposition in Pseudogrion decorum. Orthetrum s. Sabina and Neurothemis intermedia were studied on account of the reproductive ethobiology by Sengel, Bhandari and Saxena, 1994. Mitra, 1996, dealt with the reproductive behavior of Pseudogrion rubriceps and detailed studies on reproductive behavior of Seriagrian coromandelianum, Ischnera aurora, Orthetrum neglectum, Orthetrum sabina at Asan Reservoir, Mitra, 1998. The reproductive behavior of Anax parthenope, Seriagrian coromandelianum, Dysperonera quadrimaculata, Ischnera aurora, Neurobasis chinensis, Orthetrum sabina, Orthetrum glaucum, Orthetrum pruinosum neglectum, Pantaliflavicins and Pseudogen rubriceps has been studied in details at Holbaha Dam, Punjab, Bangalore, Karnataka and Aravalli Range and Desert Ecosystem of Rajasthan by Sharma, 2005, 2009, 2010a and b, 2011, Sharma et al. 2008, Sharma and Vashistha, 2011. Though, the generalized patterns of these reproductive behavior are quite same among the world of dragonflies, till detailed study reveals that they vary greatly from species to species. The present investigations has provided information on different aspects of reproductive behavioral patterns of Pantaliflavicins fabricius, and duration taken during each activity, which will help in further studies to the researchers, and it also highlighted the importance of dragonflies in nature and focus on immediate actions for conservations of odonate species and especially their habitats in India.